Hello and welcome to Dunia's Alchemy. This is a GCSE Biology lesson on transport in cells. So we're going to look at all three processes along with some examples, factors that affect diffusion, surface area to volume ratio, osmosis required practical, along with some exam practice questions and answers. So let's begin. Diffusion, osmosis and active transport. We're going to look at what they are and an example of each one. Diffusion is the movement of particles from a high concentration to a low concentration. High concentration means a lot of them, and a low concentration means where there's very little of them. This is a passive process, meaning it does not require energy. An example could be perfume particles spreading, so they move from a high concentration inside the bottle to a low concentration outside. Next is osmosis. Osmosis is the movement of water molecules from a high concentration to a low concentration, and this happens across a semi-permeable membrane. You need to have these three things in your definition for osmosis. Osmosis, just like diffusion, is also a passive process, meaning it does not require energy. The reason why it doesn't require energy is because it moves from a high to a low concentration. An example of where this can happen, so if we look over here, you can see the water molecules moving across a semi-permeable membrane. So that's that divide down the middle. The water molecules can pass through, but the larger sugar molecules are unable to pass through it. Next is active transport. Active transport is the movement of particles, but this time we're moving from a low concentration to a high concentration. This means this is against a concentration gradient. This does require energy, that's why we call it active. An example of where this can happen is in the root hair cell. So if you look at the minerals, you're going to see there's very little of them outside in the soil compared to a lot of them inside the root hair cell. So there's a low concentration outside and a high concentration inside. According to diffusion, it should move from high to low. But if it did that, then all the minerals will end up leaving the root hair cell, and we don't want that to happen, or at least the plant doesn't. So what happens instead is active transport is used, so then we end up moving the minerals from a low concentration to a high concentration. We will now have a look at which factors affect diffusion. The first factor that affects diffusion is the concentration gradient. All that means is that the larger the difference, the faster the diffusion happens. So if I have a lot of particles in one area compared to very little in another area, since there is a very large difference, the diffusion will happen a lot faster, and molecules or particles are going to quickly move from an area of a high concentration to an area of a lower concentration. The second factor affecting diffusion is temperature. And as you guessed it, a higher temperature means that there is more kinetic energy in the particles and therefore they move faster. And if they're able to move faster, they will be able to cross that concentration gradient and move from a high concentration to a lower concentration. The third factor affecting diffusion is surface area. A larger surface area means faster diffusion. In the next slide, we're going to look at how that works. And last but not least is the thickness of the membrane. The thinner the membrane, the easier it is to get from one place to the other, and therefore the faster the rate of diffusion. Let's have a better look at surface area. Looking at the pictures below, you can see a lump of rock turning into powder. That means you've increased surface area. To show it in more detail, imagine you have a sugar cube surrounded by water. That's the image over here. The water only has access to area exposed, and that's shown with the red lines. However, if I was to cut the sugar cube in half, the water now not only has access to all around the cube, but also the inner surface that you've just cut. This is how increasing the surface available increases your rate of diffusion. You do this without realizing any time you add sugar cubes to tea. Using the spoon, you'd break it up, meaning increase the surface area. The smaller cube on the left would release dye into the water quicker. That's because it has a large surface area to volume ratio, meaning for its size, more of the cube's surface is exposed to the water compared to the large cube. This allows dye molecules to diffuse out faster, which is why the left cup shows more blue colour spreading through the liquid. Finally, let's look at surface area to volume ratio in a little bit more detail with some actual examples. Cells need to exchange materials efficiently, and the bigger the ratio, the easier this exchanges. Unicellular organisms are efficient at diffusion. This is because their small size compared to the surface around them means they have a high surface area to volume ratio. Therefore, they can exchange materials faster. However, multicellular large organisms like you and I need specialized transport systems because our large size means we cannot rely on oxygen to simply diffuse into our bodies, thanks to the large amount of cells we are made of. Examples of organs which take part in the transport systems specialized to allow this exchange to happen are the heart and lungs, found in the circulatory and respiratory system. We will go over these two in detail in another video. We've already looked at the definition for osmosis, but just a quick reminder, it's the movement of water particles from a high concentration to a low concentration, and it happens across a semi-permeable membrane. In our case over here, I've given you 3 beakers. Again, a very simplistic image, but the point is really, really important. So, the cell is shown as a key on the side, and those small blue circles represent water molecules. Pause the video now and write down which direction the water would move in the beakers. So I've given you some prompts below. Does water move out of the cell? Is there no net movement of water? Or does water move in the cell? Feel free to pause the video and have a think about it. So, let's have a look at whether or not we have a high concentration inside or outside the cell. In the first picture, you can see that there are more water molecules, meaning those blue circles, outside the cell. So, I'd call it a high concentration outside and therefore a lower concentration inside. Looking at our definition for osmosis, it always moves from high to low. So therefore, water should be moving into the cell. Looking at the second beaker, you can see that there are more water molecules inside the cell compared to outside. This could be done whenever you have a very sugary solution. So a high concentration would be found inside the cell and a low concentration would be found outside the cell. And according to the definition of osmosis, it moves from high to low. So in our case, it would move from inside the cell to outside the cell. So water moves out of the cell. And then lastly, again, not the best representation, but I'm sure you get the idea. You have 3 water molecules on the outside and 3 on the inside, meaning they're both the same. And therefore, there is no net movement of water. That doesn't mean water doesn't move at all. It's just that there's no overall direction where it's either leaving or entering the cell. These solutions are said to be isotonic. Let's break down the word isotonic. Iso is the prefix derived from the Greek word meaning same, and tonic means strength. Therefore, isotonic solution is one with equal strength to a cell. In an isotonic solution, water moves in and out of the cell at the same rate, essentially the same concentration inside and outside the cell. This results in no net movement of water. This was the correct answer. So our last beaker is an example of an isotonic solution. When dealing with high water concentration outside the potato, know that this can be referred to by your exam boards as dilute, and one with a lower water concentration or high sugar concentration could be referred to as concentrated. These are the steps for the osmosis required practical. Feel free to pause the video, this should take you around 7 minutes, to put these in the correct order. Let's have a look and see if you got all of them correct. The first step is you label the beakers with different sugar concentrations. Your concentrations can range from 0 to 0.2, 0.4, all the way up to 1. Second is you cut equal sized potato cylinders using a cork borer. Then you measure and you record the initial mass of each cylinder. You place each potato into different solutions. Make sure the equal sized potato cylinders are put into all the different solutions, and then you leave them there for a set time. Once that's done, you gently remove or blot dry to remove surface water. You measure and record the final mass of each potato. Repeat for reliability and calculate the mean. Calculate the percentage change in mass for each concentration. And then finally, plot a graph and identify the isotonic point. Remember that word isotonic means same strength. Let's answer an exam style question together. A student investigates osmosis using potato chips. They place chips in different concentrations of sugar solution and they measure the percentage change in mass after 30 minutes. So after the actual practical has been carried out, here's their results. Part A says, identify the sugar concentration at which the potato is isotonic. If you remember, isotonic means same strength. So since it has the same strength, we're not expecting any net movement of water into or out of the cell. So therefore the percentage change in mass shouldn't change much. And that's how we would say the sugar concentration of 15 because the percentage change in mass was at 0. Part B. Explain why the potato gains mass in 0% sugar solution, but it loses mass in 30% sugar solution. This goes back to the point we looked at earlier with our beakers. 0% sugar solution means basically no sugar in there, it's all water. So that is an extremely high concentration of water that the potato is surrounded by. So if it's surrounded by a lot of water, the water will move into the potato. It should gain mass. However, at 30% sugar solution, the potato has more water than the actual solution, so water would move out, and therefore it loses mass. And lastly, suggest one factor the student should keep constant to ensure a fair test. And you could say either same volume of solution, same size of potato chips, same duration of experiment, or same temperature. Any of them are valid for that mark. And over here, I've left the answers for all three. Doing practice on these questions is really important. So I've left a whole bunch of questions over here, bronze, silver and gold. These are to help check your understanding of the topic. This should take you 20 minutes, so feel free to pause it, and the answers will come shortly. Here are your answers for transport in cells practice questions. Make sure you mark them. Gold will be on the next part. Right now you can see bronze and silver. Make sure to give it a mark out of 21, and in a different colour pen ideally. Make sure you add in the correct one for anything that you missed out. And here's your answers for gold. Did you get all of them right? Let me know in the comments below. This is our video complete. We have now gone over the 3 processes along with some examples, different factors that affect diffusion, surface area to volume ratio, osmosis required practical, and some exam questions and answers. So this was pretty loaded, and I hope it really benefited you. Thank you for watching, and make sure you do the 3-2-1, share it with 3 others, 2 clicks, subscribe and like, and finally 1 comment. Have a great one.